Now we're going to look at two opposing processes. The opposing processes to glycolysis, that would be gluconeogenesis. Glycogen is the major storage form of glucose in mammals, and we have glycogen at the top of our chart here. We're going to look at both the breakdown and the synthesis of glycogen. We will start with glycogen synthesis. However, we can see from this flow chart the connections to other pathways. So for example, glucose can be converted to glucose 6-phosphate, or we can have the reverse reaction. We can break down glycogen to get glucose 6, or we can use it to produce glycogen. Glucose 6-phosphate can be converted to ribose 5-phosphate and enter the pentose phosphate pathway. Sorry about that. Excuse me. And glycolysis produces pyruvate, while gluconeogenesis utilizes it. Pyruvate can be formed from amino acids or utilized to form amino acids. Pyruvate can also be converted to lactate and vice versa. And of course, we know that pyruvate can be converted to acetyl-CoA and enter that citric acid cycle. Before we look at gluconeogenesis, I want to take a look at what is called the Cori cycle. And we have seen that under anaerobic conditions, pyruvate is converted to lactate. And the following diagram describes this cycle of lactate conversion to glucose. The Cori cycle is named after its discoverers, Carl and Gertie Cori, who studied glucose metabolism at Washington University in St. Louis. This cycle, or their cycle, describes the process that occurs when glucose is converted to lactate during intense exercise, and then lactate is converted back to glucose during a recovery period. So the cycle is initiated by anaerobic glycolysis during exercise. So if we are exercising strenuously, we are using glucose in our muscles. Our muscle cells are metabolizing glucose, and we begin to produce high levels of lactate. Most of the lactate is released from the muscle and travels in the blood to the liver. So we've got muscles, we have glycolysis, we produce lactate, and this lactate is then carried in the blood to the liver, where gluconeogenesis occurs. The remainder of this, the lactate is then converted to glucose via gluconeogenesis that occurs in the liver, and then that can enter into the bloodstream as well, back to that respiring muscle tissue, and undergo another round of glycolysis. So what is gluconeogenesis and how does it operate? Genesis meaning generation, neo meaning new. So basically we are forming glucose from non-carbohydrate precursors. Metabolism in humans consumes about 160 grams of glucose per day. 75% of that is metabolized in the brain. Body fluids contain only about 20 grams of glucose, and the glycogen stores, which we're going to look at the production of glycogen momentarily, only store about a 1-day supply of glucose. So 180 to 200 grams of glucose are stored as glycogen. These stores are typically depleted during strenuous exercise, so we have to have another way to make glucose. And that's what we're going to look at right now. So This is just a reminder. This is a slide we've looked at before about regulation. We're going to focus on B. In glycolysis, we know we have 3 major regulatory sites: reaction 1, 3, and 10. So let's pretend this is glycolysis. Here we only have 1 regulation point, but we do have 3 in glycolysis. So there are 2 different enzymes that regulate the forward and the reverse reactions. And that is exactly what we're going to see in gluconeogenesis. There's 3 sites, and those are those regulatory sites that use alternate enzymes. The remainder of the enzymes, however, are shared with glycolysis. So here's a look at that pathway for gluconeogenesis, which is going from bottom to top, glycolysis from top to bottom. So as I said, there are 3 regulatory sites or 3 enzymes that have to be changed or regulated in order for gluconeogenesis to occur. And we're going to start at the bottom. The steps, though, that are bypassed are hexokinase reactions, phosphofructokinase, and pyruvate kinase reactions. So in gluconeogenesis, we are going to start with 2 molecules of pyruvate and convert that to 1 molecule of glucose. Please note, however, that we can use, as we've just seen, lactate can be converted to pyruvate. Also, some amino acids can be converted to pyruvate as well. And remember, 90% of gluconeogenesis occurs in the liver, about 10% in the kidneys. So the first reaction actually requires 2, or the reversal of pyruvate kinase requires 2 enzyme catalyzed reactions, each of which requires an ATP equivalent. In the first step, we are actually using ATP, and the enzyme responsible for this reaction is pyruvate carboxylase. So we need also bicarbonate and ATP for this reaction to occur. So in this step, we are converting pyruvate to oxaloacetate. So we're adding a CO2 or a carboxylate group, and we are using the energy from the hydrolysis of ATP to drive this reaction. So here we have oxaloacetate. We could also utilize this reaction if we need oxaloacetate in the citric acid cycle. So here's another source of oxaloacetate for TCA. Second step, if we're proceeding with gluconeogenesis, utilizes GTP. So this is an equivalent energy molecule just like ATP, guanine triphosphate. The enzyme here is phosphoenolpyruvate carboxykinase. Kinase. What does that mean? Kinases, if you remember, transfer phosphate groups. So we have GTP gets hydrolyzed, and the kinase is going to attach that phosphate group to produce phosphoenolpyruvate. We also remove a molecule of CO2. So we have a decarboxylation of oxaloacetate and the phosphorylation then of the remaining molecule. So both of these enzymes are going to be regulated. Regulation of pyruvate carboxylase is allosterically regulated by increased concentrations of acetyl-CoA or decreased concentrations of ADP. So that would mean if we have, well, we'll come back to that later. PEP carboxykinase is also regulated by low concentrations of ADP. So once PEP is formed, the next 5 reactions basically just proceed in reverse until we get to fructose 1,6-bisphosphate. And where are we here? So once we get to fructose 1,6-bisphosphate, then we need another enzyme to bypass that regulatory step. So in order to convert fructose 1,6-bisphosphate to fructose 6-bisphosphate, we have the hydrolysis of one of those phosphate groups, and actually it's the phosphate in the 1 position, so we have a release of inorganic phosphate. So in the forward direction, or in glycolysis, we have a kinase. So again, that kinase is going to transfer a phosphate group to fructose 6-phosphate, and in the reverse direction, we are going to dephosphorylate fructose 1,6-bisphosphate to produce fructose 6-phosphate. So how is this regulated? Yes, we just looked at enzymes that are utilized to bypass those regulatory steps, but we have to have some other mechanism to control these 2 pathways. So in glycolysis, or the catabolic direction, this process generates a net of 2 ATPs. In gluconeogenesis, tongue-tied today, the anabolic direction, there are 6 ATP equivalents used up, so this costs us energy. If these processes or 2 pathways were uncontrolled and were able to simultaneously function, we would just burn up ATP without producing any kind of useful work. So we have to regulate this such that glycolysis proceeds when ATP concentrations are low, and gluconeogenesis occurs when ATP supply is insufficient. Or is sufficient, sorry. The enzyme responsible for this is fructose 2,6-bisphosphate. This is a potent stimulator of phosphofructokinase in glycolysis and a powerful inhibitor of fructose 1,6-bisphosphate in gluconeogenesis. Okay, so this is a slide I put together that hopefully will help you visualize the kind of regulation other than the enzyme regulation that we've just talked about. Concentrations of metabolites will also affect these pathways and their regulations. So for instance, well, let me first start by saying anything that is red is going to inhibit that pathway. Anything that is green is going to activate that pathway. Okay, so let's just take a look at glycolysis first. If we have low energy, or high energy, we have high energy. That means we have high concentrations of ATP, and we don't need to make more. If we have high concentrations of ATP, we'll turn off glycolysis and we will turn on gluconeogenesis. Okay. Or we will produce glycogen. Actually, we'll produce glycogen. If our AMP concentration is high, that means we have low energy. So we want to turn on glycolysis. Similarly, fructose 1,6-bisphosphatase will activate high levels or citrate, high levels of citrate are going to increase or turn on gluconeogenesis, and so forth. So make sure you take a look at this and understand why and how these metabolites are affecting the regulation of both these processes.